Hi everyone, welcome to the last video of the course. We'll discuss a couple more interesting studies, I think, about how evolution experiments are done in lab and what sort of light do they shed. And then I will discuss a couple of broad problems that we haven't had a chance to discuss in this short course. So, last time we discussed how synonymous mutations alter just because. Let me just briefly summarize what we did, that synonymous mutations can have fitness effects. This is not to say that every synonymous mutation is going to have a fitness effect. Synonymous mutations can have fitness effects, and they do this by one, changing protein amount, and this protein amount can be changed via several different ways. First, by altered stability of mRNA, as a result, if stability decreases, that means you have lesser mRNA and you get less protein. Secondly, you can get that by altered access to ribosome. If at the 5' end of this mRNA we have a tight hairpin or ribosome binding site is embedded deep in the mRNA structure, then it is harder for ribosome to get access. And lastly, that use of a rare codon which slows down translation. Secondly, and somewhat surprisingly, synonymous mutation can also change protein structure. And this we saw was because of an altered co-translational folding kinetics. And we for both these studies we know of several examples which illustrate this point. And again I want to emphasize that is not to say that every synonymous has a fitness effect, but many synonymous mutations do have a fitness effect. In a study that was done a few years ago, it illustrated this point very nicely. What they did was, you have DNA and then transcription takes place and you get mRNA, and this mRNA is synthetically created in such a way that the first 3/4 encodes for GFP, that means the green fluorescent protein, but not the full GFP, let us say only 3/4 of the GFP. Then you have 1/4 you have the 1/4 remaining part, and after that you have RFP, which is red fluorescent protein, but not the full RFP, but 3/4. And now what the authors did, that in the first scenario you place rare codons here. After the 3/4 GFP you place rare codons. And what these rare codons do is that when ribosome comes here, there is a long pause because it has to find the appropriate tRNAs which are carrying these amino acids which are being encoded by these rare codons. As a result, the GFP amino acids, the 3/4 GFP amino acids that have been encoded here, they will not stay like this, they will fold and we get a protein structure which is encoded by 3/4 of GFP. And this does not fluoresce. So, this is GFP, but not full GFP, and it does not fluoresce. Eventually, these rare codons are being translated, and then the rest of the transcript is translated fast, and this remaining bit when it is translated. So, now we have this GFP 3/4 GFP and the rest of the codons which are encoded by this 1/4 of GFP and 3/4 of RFP. Now, interestingly, this 1/4 is such that it will fit in with GFP and make the GFP complete, or this 1/4 can also fit in with RFP and make the RFP complete. So, in this particular design, when rare codons are here, GFP folds and this 1/4 is then unable to incorporate into this already folded GFP structure. So, what happens post excuse me, what happens now is that this rest of the amino acid chain folds and we get the following structure, that we have a 3/4 GFP and a fully folded RFP. Excuse me, and 3/4 GFP. So, because this cannot fluoresce, when I look at the cells, the cells are fluorescing red. So, when I look at these populations under a microscope, the cells will appear as if they have red fluorescent protein, which is being made because of these following reasons. However, the authors of this study do this altered design, where now we have this DNA, the resulting mRNA. This is the part which is for 3/4 GFP, then we have 1/4 GFP, and then we have 3/4 RFP. Now, instead of placing the rare codons here, the authors place the rare codons here, which means that when this transcript is going to be translated, the ribosome halt is going to come when it is here, and the first part of the transcript is going to be translated reasonably fast. As a result, the amino acid chain is going to be now that the translation process has halted, this amino acid chain will now fold, because there is a long pause when ribosome is not moving. But this 1/4 GFP will now work with the 3/4 GFP and form the fully folded GFP. So, this is full GFP, because this 1/4 is now working with this 3/4 GFP. And once these rare codons, these are the rare codons, once these rare codons are translated, then translation resumes and the rest of the protein is then translated, and when the ribosome comes here, this is the rest of the protein. And eventually this will translate, and the net result of the folded structure would be this, where this is 3/4 RFP, and this of course will fluoresce as green. And all of this, interestingly, all of this is accomplished by only changing the location of where these rare codons are. In the previous case, you place some rare codons here and you get a different shape of the protein and the cells are fluorescing red. And in this case, you change the position of the rare codons to after the 1/4 part, and in which case the cells are fluorescing green. So, these synonymous mutations have this ability to not just change the protein amounts via these different mechanisms we discussed, but also protein shape, and protein shape is eventually what decides protein function. Hence, a synonymous mutation can change protein function also. This I think is a extremely interesting result to have come out in the last few years. All right, let us quickly also discuss one example of what happens in a mutation accumulation experiment. That is the experimental technique which we did not quite discuss after we discussed how it is performed, but what is the sort of an observation that comes out of a mutation accumulation experiment. So, let us imagine that we have E. coli. The way we will do these experiments is that I will grow them in a tube, then I will take a very small volume V naught, and this V naught, let us say, only contains about 100 individuals. I will spread these 100 individuals on a plate. Of course, I can't see individual E. coli, but when I spread them, my calculation shows that this much volume of the liquid must have 100 individuals. So, when I spread them, I know there are 100 individuals there, but I do not know where are they. So, they will be spread somewhere whose location I do not know. So, there are these 100 individuals spread over a Petri plate. What I do, I want to pick one of them, but I do not want to make any biased choices. So, when I wait for, I let this grow overnight in a incubator, grow overnight at 37, and what will happen is that for each one of these individual cells that was placed on a Petri plate, it has access to resources and temperature is right. So, doublings will start to happen, that one individual will become 2, 2 will become 4, 8, 16 and so on and so forth. By the time I give it 14 to 16 hours, these would be many cells and they would look as little mounds like this on a plate, and each of these mounds is called a colony. Now, obviously, in this process mutations have happened, and I want to pick one individual from one colony completely randomly. Now, how do I randomize this process? Remember, mutation accumulation experiments are where I want selection to be absent. So, everything has to be completely randomized, only drift has to decide the fate of this evolution experiment. So, how do I do this? So, the typical technique is that I do not wait till this moment to pick an individual colony. That is because by looking at the colony, I will be biased. So, some people like to pick a very average looking colony, some people like to pick big colonies, some small and so on and so forth. Some colonies might appear different color, shape and so on and so forth. This is all happening because of the phenotypic state of the cells in that population and the mutations that have been acquired. So, you are attracted to a certain type of a colony, and in order to sort of get rid of that bias, what we do at the time of plating is that I will mark a section of the plate completely randomly. So, I do not know where these 100 cells are, but at the time of plating I will physically mark a part of the plate like I have shown here. And the idea being that I am saying that whichever colony comes inside this box or closest to the box is one that I am going to pick. That might be the biggest, smallest, colorful, irregular shaped, most regular looking, any colony. I am not going to care for any of that, but whichever colony comes here is the one that is going to get propagated in the experiment. Everybody else is going to get discarded from the experiment. So, in this way, this choice of picking this colony was randomized. Now, the choice of colonies has been randomized, but now I want to pick one individual from this colony completely randomly. I do not want to pick the cells at the periphery, I do not want to pick the cells near the center of the colony, at the top of the colony. I want to pick every cell that is present in that colony should have an equal chance of being picked and propagated forward. So, what I do to ensure that is that I pick this colony up physically from the plate and I put it in a culture tube and vortex it such that all members of the colony are spread in this tube here. Now, I take V naught amount from this. So, when I take V naught amount, my goal is to take about 100 individuals from this colony. And now, because every individual has been picked up from the plate and suspended in this tube, and out of that entire population I am going to pick 100 individuals. Every individual has the same probability of being picked and every individual has the same probability of not being picked. So, this choice of which 100 individuals I picked from this colony in this way has also been made completely randomly. I did not just simply touch the periphery of the colony and suspend it. I tried to pick as much of the colony as I could. Of course, there will be some cells which will be kept there, but that is trivial as compared to what I picked. So, this has also been randomized, and after picking this I do the same step again, that I spread this on a plate, and again I do not know where are these 100 individuals because I can't see individual cells, and at the time of plating I am also marking a area in the plate where I am saying that whichever colony comes inside or closest to this particular marked area is the one that is going to get propagated forward. So, in this way the choice of colony and the choice of 100 individuals that are being propagated in the population is completely randomized. Now, when I do this, what is going to happen is that this colony, every colony has 10 to the power 8 cells. That means lot of mutations have taken place here, and there will come a time that by chance these 100 individuals will also have individuals which have acquired mutations, and eventually I will end up picking a colony which was founded by one of those individuals which was already carrying a mutation, and so on and so forth. So, as I move forward and do more and more plating, more and more mutations will get accumulated in this line of experiment, and hence this is called mutation accumulation. I forgot the word accumulation, mutation accumulation experiment. We are accumulating mutations in a line, but without letting selection act. So, what is a typical sort of readout from this experiment is that if we look at this process is called one transfer. And a typical mutation accumulation experiment, you will do this maybe 200 times, 300 times. And as we saw with Lenski's experiment, you will not do only one line, you will do several lines in parallel. Let us say you do 100 lines in parallel of like this, and you are doing this parallel work for 200 transfers. So, if that is the case, then what is the sort of a readout? One interesting readout is that I would like you to plot the following. Let us say this is number of transfers, and this is fitness. Let us say this is the starting fitness F naught, which is the ancestor fitness. What I would like you to try and guess here is that how would the fitness change as I go do this experiment for, let us say, 300 transfers. I will give you 10 seconds to think about it, and then we will discuss what is actually seen in a mutation accumulation experiment. So, let's normalize this ancestor fitness to 1. What is seen in all mutation accumulation? Mutation accumulation experiments has been done on a variety of organisms, from C. elegans to E. coli to yeast to Drosophila, and every one of them shows this particular pattern, that as populations are propagated, this is what happens to fitness. Fitness decreases almost linearly. And remember that we said that we had eliminated selection from this experiment. So, if we had eliminated selection, why is fitness decreasing? It is decreasing because as this individual is being evolved in the population, let us say this was ancestor genome, after going a further ahead, it accumulated one mutation. After going a little bit ahead, it accumulated another one and another one, and so on and so forth. So, as it is moving forward, it is accumulating more and more mutations. Now, given a mutation, what is our null expectation? That given any random mutation, is it more likely to be beneficial or deleterious? And I think we all recognize by intuition that deleterious mutations are more likely to occur as compared to beneficial mutations. So, as this line is moving forward, it's more likely to acquire a deleterious mutation as compared to a beneficial mutation. I mean, some beneficial mutations will also get acquired, but by and large more deleterious mutations will get accumulated. As a result, this fitness of this line, when I check it periodically, will be continuously decreasing. Now, what I can do, so that is one, which is basically just a consequence of the fact that there are more deleterious mutations as compared to beneficial mutations. What I can also do is that I can sequence these lines and I can try and find out what is the nature of mutations that are happening, how many are SNPs. So, I will sequence this, I will sequence the ancestor that I started with, and I will compare them and find out that what is the nature of mutations, how many are SNPs, how many are insertion deletions, how many are duplications, and so on and so forth. So, I will quantify this, and this gives me a very detailed information of not just about what is the mutation rate, but what is the mutation rate associated with each one of these types of mutations. So, that is another important piece of data that comes out of these mutation accumulation rates experiments. And obviously, I do not do this in one line. We said that we are doing each line for maybe 300 transfers, but we are also doing 100 lines in parallel, just as Lenski did 12. So, each one of these lines is going to give me one trajectory. So, I do not have one line here, I actually have 100 lines here. Mutation accumulation experiments conclusions are never drawn from one line, because mutations are occurring randomly, and it is possible that by chance one line could acquire more SNPs and may have no duplication, whereas another line may have more indels and very few SNPs and duplications. But when we average these effects over 100 different lines, then we get a statistical measure of what is actually happening in the genome of the organism. Fine. So, that is the sort of inputs that we have, that we obtain from mutation accumulation experiments. And as I said, a number of organisms, these type of experiments have been done for a large number of organisms, and the lessons that we draw for them are robust. The principles hold across organisms of different complexity, and a lot of what we know about mutation rates actually comes from studies such as this. In the last few minutes of the course, I want to discuss a couple of aspects that we haven't had a chance to discuss. The first one is to do with the fact that ecological niches rarely have one species. So, ecological niches have much more than one species. In fact, there has been a lot of study. So, what are the popular niches that people study? Isolating. So, one of the popular ways of studying how bacterial species interact with each other is through studying bacteria that are present in soil. And there have been studies which have tried to isolate different species from a very small grain of soil present in a garden, for instance. And what we find that even in a very small grain of soil, the number of species that are present is in the several dozens. So, this particular sample from a study that came out about 10 years ago, it had 4 to 5 dozen different species, 50-60 species coexisting in that grain of soil. Our group has isolated bacteria from soil, and from 1 gram of soil we isolated 25 different bacterial species that were co-inhabiting that niche. Remember, these are species that we have been able to isolate. There are many others that we have not been able to isolate. So, the number of species which are coexisting is very, very large. Now, this poses a few questions, that why is the fittest not taking over? What are the principles that maintain this population structure? And this could be thought of from the context of soil microbiome or even gut microbiome. So, lot of studies take place these days which are studying the microbiota that is present in our intestines and how is it that species dynamics changes. The second issue is of stability, that these systems are exposed to perturbations all the time. In soil, sometimes it floods, sometimes it is dry, temperatures are high during the day, temperatures go cold at night, and so on and so forth. We drink a lot of water, we eat, but then we do not eat for hours, and so on and so forth. So, how is it that these niches have these dozens of interaction species, but they maintain their numbers, they do not go extinct? How is stability or robustness of these niches maintained? So, these are two important questions on which a lot of work is going on these days. The last thing that I want to discuss is the phenomena called HGT, which is called horizontal gene transfer. The idea of evolution that we have discussed in this course is that I as an individual divide into two progeny, and if division is faithful, then the progeny have identical DNA, but then somewhere down the line one of the progeny will have a mutation. So, evolution in this sense is vertical, that evolutionary change takes place across generations. As you transition from one generation to another, evolutionary change can take place. A few decades ago, it was found out that actually, so in this vertical view of evolution, what is true is that individuals do not evolve, species evolve across generations, because if I have a certain genotype, I am going to live with that genotype and not evolve, but my progeny might have a different genotype which might be better suited for the environment that we are living in. So, I as an individual do not evolve in a biological sense at least, and species evolve through generations. That is the understanding that we have through this vertical prism of evolution. As against this, a few decades ago it was found out that actually bacteria have this phenomena that if they are carrying pieces of DNA, they can do trade of DNA where this individual can actually acquire the little piece of DNA from that individual, and this trade of DNA can happen. And the popular mechanisms of this is direct trade with another bacteria, or viruses carry pieces of DNA around. So, one virus might have infected another individual and is carrying its piece of DNA, and when it infects me, I inherit that piece of DNA. And third, from the environment. So, many species, the most popular one that studied is a soil bacterium called Bacillus subtilis, have this capacity that if in the environment there are pieces of DNA, it has the ability to pick up a piece of DNA and become this. So, through horizontal gene transfer, I do not need to divide and I am able to evolve as it is. So, this is horizontal because I can trade genes with my peers of my own generation and evolve in a biological sense. And this has huge evolutionary implications and very interesting and dynamic mode of evolution, because remember here the kind of changes that are happening are small. There was a G here, there was a T here. So, this is in most cases this leads to small incremental change. There was a protein functioning, you made it better. But in this particular case, you can acquire completely new traits because you are acquiring large pieces of DNA from the environment through any of these three mechanisms and evolve. Again, lots of work being done to understand this, lots of experiments, interesting cool experiments that are being done to understand this. But for the purpose of this course, we stop here. I hope you have enjoyed these 30 hours, and I hope the sense of these 30 hours is to just scratch the surface of this beast of a subject that we call evolution. And those of you who are interested, I hope you will take up further courses to enhance your knowledge in the area. Thanks a lot, and we close the course with this.